The overall goal of this procedure is to demonstrate how to assemble and use a dual labeling DNA damage sensor that labels two major types of DNA breaks in tissue sections. The sensor is comprised of a 30-tumer DNA oligonucleotide, dual labeled with FITC and rhodamine, and an enzyme, vaccinia topoisomerase 1, or VAC-topo, that self-assembles when VAC-topo binds to the 30-tumer. The machine operation starts when topoisomerase cleaves the oligonucleotide at the 3' end of the recognition sequence, resulting in a VAC-topo-activated FITC-labeled unit and a rhodamine-labeled free hairpin with a terminal 3' hydroxyl. The units are short-lived and quickly reassemble back into the original construct, which is subsequently recleaved. In the absence of additional extramolecular DNA breaks, these two units continuously separate and religate in a cyclic manner. When the molecular sensor is applied to tissue sections with DNA damage, the FITC-labeled detector unit selectively attaches to blunt-ended DNA breaks with 5' hydroxyl. The rhodamine-labeled detector unit will ligate to a 5' phosphate blunt-ended break if T4 DNA ligase is present. Results are obtained that show the presence and distribution of DNase 1 and DNase 2 type DNA breaks in cells based on fluorescence microscopy.
The main advantage of this technique over existing methods for apoptosis detection, like tunnel, is that it is more specific and provides additional information by labeling both self-autonomous and phagocytic phases of apoptosis. From the nanotechnology perspective, the sensor exemplifies